Did you really?
Assessment that I filled out.
Hello, everybody. Can you hear me?
Yeah, we can.
Somebody?
Yes.
Anybody?
Yes.
Okay, thank you. I never know. I don't get any feedback, so I can't tell if you're hearing me. Today, we're going to finish talking about HIV. We're going to pick up where we left off last time, and then we're going to start talking about the genetics of cancer. And we'll go through that in some detail. We're also going to talk a little more detail on RNAi, how it works, things like that. Why it's called a knockdown and not a knockout. And I have been recording all of the lectures through Zoom, and I'm hoping sometime today to have them all uploaded to YouTube. I will send you the information regarding what to look for, but basically it's going to be Bio 324. And then there's going to be a descriptor of what the lecture is about. Now, one lecture in particular, there's about 10 minutes of dead time at the start, because I logged in too soon and it started recording. So I need to go back and figure out which one that is, and I'll tell you in an email when I send you the information regarding how to access it. We've been talking about HIV, which doesn't get near the press time that it once did. The coronavirus has pretty much taken over everything. And well, it should, because it's far more contagious than is HIV. I think I told you, if you want to read about the early history of HIV and the gay lifestyle in San Francisco, there's an excellent book called And the Band Played On, just very well written. The author was a journalist in San Francisco who was part of the gay community, and he eventually died of AIDS from it. We talked about how the virus attaches itself, CD4 receptor, on our cells, and the virus has a glycoprotein, GP120, which interacts with the receptor. In doing so, it causes conformational change. The membrane protein and whatnot shifts somewhat, and it ends up recruiting other receptor proteins. And one of the other receptor proteins, at least on the most common form of HIV-1, is the chemokine receptor 5, CCR5. It's found on the surface of white blood cells. It was discovered in, I believe it was San Francisco again. There was a gay couple who did not practice safe sex. One was infected with the HIV virus and ultimately died of AIDS. And in spite of numerous exposures, the partner never came down with the infection. And they found out that he was actually homozygous mutant for this particular receptor. So the virus couldn't get into the cells. Okay, come on. Yeah, some people, as I say, are mutant for this particular receptor. Some of the majority of people are heterozygous, but there are some individuals that are homozygous mutant. Being heterozygous confers some resistance to infection, but being homozygous is pretty much a guarantee that you're not going to be able to pick up the most common version of HIV. CCR5, it's a delta 32, it's a deletion of 32 bases. It protects, historically, individuals from both smallpox as well as the Black Plague, Black Death, but increases sensitivity to other viruses like the West Nile virus. As you might expect, the populations where you see this deletion most common are those that were subjected to the selection pressure of smallpox and plague. It's estimated that between 4 and 20% of people of European ancestry are heterozygous for this deletion. That's not surprising given the incidence of both smallpox as well as the plague in Europe, particularly Northern Europe, that there would be strong selective pressure to, much like sickle cell anemia and malaria, to select for individuals carrying this particular mutation. Highest incidence is in people of Nordic ancestry, Northern Europe. The frequency declines as you move further south, as did the incidence of smallpox and plague. It was thought one time that they would be able to use RNAi to knock down the receptor CCR5, and we'll talk about that in a couple of minutes. Okay, that's just a quick overview of what we ended on last time. Pharmaceutical companies spend a ton of money investigating RNAi as a potential therapeutic. And we'll talk about why it was not successful a little bit later on in the lecture. RNAi is an RNA-dependent gene silencing, but unlike CRISPR-Cas9, it targets RNA and not DNA. CRISPR-Cas9 targets DNA, RNAi targets transcribed RNA. It's a post-transcriptional regulation of gene expression using double-stranded RNA initially. As I said, a lot of money was spent on developing RNAi libraries. A lot of money was spent on trying to develop these as a therapeutic agent. But unfortunately, one of the characteristics of RNAi is that a very high incidence of off-target effects. And we'll talk about why that is in just a couple of minutes. RNA interference, RNAi. It knocks down endogenous levels of RNA or protein that is derived from the RNA. It does not eliminate it. CRISPR-Cas9 can totally eliminate the gene. RNAi simply reduces either the RNA itself or the protein product. In the end, the protein product is going to be knocked down or reduced in levels. It involves something called a RISC complex, RNA-induced silencing complex. The mRNA, if complementation is perfect, that is the RNA directing RNAi is a perfect match with the target RNA. Most of the time, especially in plants, it causes degradation of the messenger RNA. In situations where there's only partial complementation, only part of the sequences match, it's going to cause inhibition of translation. And this is where the problem comes in terms of it being a therapeutic agent. It does not require a great deal of complementarity for the RISC complex to bind and inhibit translation of RNAs. And it's been shown that off-target effects are so widespread in RNAi that it really has never been developed as a successful therapeutic agent, which is too bad. As I say, RNA-induced silencing complex, RISC. It's initiated by a ribonuclease known as dicer. It's going to dice up the RNA into short segments, about 20 to 25 base pairs in length. It targets double-stranded RNA. And in research labs, we basically make double-stranded RNA by a stem and loop structure, transgenic constructs that will transcribe and form a stem and loop. And it's the stem part that is double-stranded RNA and will be targeted by the protein dicer. Typically, it leaves a few bases unpaired at the end of the molecule. The fragments of RNA are referred to as small interfering RNAs, siRNAs. In my opinion, we have too many different names for different RNAs, but somewhat descriptive. It becomes separated. The double-stranded RNA becomes separated into a single strand. And at least last time I looked, nobody is entirely clear how the RISC complex decides which of the two strands to incorporate into its being, but it does so. Then the complex, the RISC and the RNA, guide RNA, will base pair with the target RNA, and in lots of cases, off targets, and either degrade it, the RNA, or inhibit its translation. As I said, it's the inhibition of translation where the problems really come in and why it's not been a very good therapeutic agent. Here's an illustration. You have double-stranded RNA This is a naturally occurring phenomenon in eukaryotic cells, both plant and animal cells. It's used in a lot of different situations, including establishment and maintenance of heterochromatin. So it plays a significant role in gene regulation, normal gene regulation. Dicer comes along, cuts up this double-stranded RNA, and then one of the two strands is chosen to form part of the RISC complex. It's the antisense, which is going to be complementary to the targeted RNA, messenger RNA. And if pairing is perfect, it's going to cause degradation of the target RNA. If it's imperfect, it's going to inhibit translation. The ribosomes aren't going to be able to utilize the information stored in the messenger RNA. This is just a different illustration. Again, Dicer chops up the double-stranded RNA, producing siRNA, short segments. Then it associates, the RNA associates with the RISC complex, allowing the RISC complex to target the messenger RNA, the target messenger RNA. But again, unfortunately, it also causes inhibition of translation. Here's HIV. Here we see CCR5, CD4, the two receptors that we've talked about, being necessary for binding and ultimate absorption of the virus, where its membrane associates with our cell membrane, lipid to lipid, and allows release of the capsid, which contains two intact RNA genomes, and each one of the RNA genomes has a reverse transcriptase attached to it. So you get infection, you get reverse transcriptase, you make DNA, the DNA then gets inserted into our host, our genome. Independent of that, the receptors, CD4 and CCR5, are going to be transcribed, go to the cell surface, provide another access to what's going on. Viral mRNA, simply going to be transcribed, and the proteins are going to be translated. So originally they thought, RNAi is going to be fantastic. Here's all the potential targets of RNAi. You could target the invading RNA strand. You could target genes that are encoded for the different proteins required for maturation of the virus. Target the pre-mRNA for CCR5, the co-receptor. You could target the mature messenger RNA. And so there was thought there's a whole ton of potential targets, but unfortunately did not work out. Too many side effects, off-target effects. We actually, several years ago, probably about a decade ago now, we had a job candidate that thought she was studying meiosis and why meiosis fails fairly often as people age, particularly females. The incidence of Down syndrome goes up significantly in females over the age of 40. It also goes up in males, but males have to typically be like 60 years old, much older than what happens with females. So she was trying to investigate what was going on. She had made an RNAi construct of a gene that she suspected was involved in meiosis, the final stages of meiosis. And that's what her job talk was on. During her talk, though, she was honest enough to admit that somebody had a knockout, which is essentially CRISPR destruction before CRISPR was such a thing, before we discovered it, where you actually destroyed the target gene. And studies from the lab that did this showed that the knockout, totally missing gene, had no effect whatsoever on meiosis. So what the person who interviewed with us was actually talking about was an RNAi artifact. She didn't say it, but the evidence was very compelling. Because a knockout should, if anything, have a much more exaggerated phenotype than simply a knockdown, an RNAi knockdown. Okay, talked about CRISPR-Cas9. They've tried to use it to eliminate HIV once it's inserted into the host genome, into our DNA. Had very limited success initially because the mode of replication of HIV was so rapid, simply transcription. It was way too fast for CRISPR to keep up with. The RNA just overwhelmed the system. They've had greater success doing sort of a double targeting. They use CRISPR-Cas9 and the cocktail, the drug cocktail that targets HIV. The analogs of RNA, the non-analogs that inhibit the reverse transcriptase, as well as genes that inhibit the protease. And they found that if they used that cocktail in combination with CRISPR, they had much greater success in eliminating the HIV virus. So it holds potential for a treatment. As I say, the drug cocktail prevents maturation of the RNA. Even though it may still be transcribed, it's not going to be able to form a mature and infectious virus. This allows for CRISPR-Cas9 to target and eliminate the genomic sequences derived from the HIV infection. Holds promise. This happened, was reported about 2 years ago. I haven't heard any updates. I'm guessing there may have been some complications. It may not be 100%. You're looking for a cure, not just something that's going to temporarily knock down the virus. Oops. Sometimes my PowerPoint burps. Recently, CRISPR-Cas9 also has off-target effects, but they're not nearly as common as RNAi off-target effects. In the old system, we talked about CRISPR-Cas9, how it, in the wild type, functional, it carries 2 copies of a nuclease, 1 for each strand of DNA, chopping up the targeted gene. And then to get replacement, you relied upon homologous recombination. So you would co-inject CRISPR-Cas9 as well as the DNA sequences for the corrected gene, the changes you wanted to make. And just hope that by luck, the small fragment of DNA that you've included will find the targeted gene and through homologous recombination will mend it. Now, more recent development is to include an RNA copy of the corrected sequence, whatever it is that you want to replace, as part of the CRISPR itself. So the CRISPR is not dependent so much on just random luck, sequences finding the cut gene, but now the CRISPR system is actually delivering a copy of the corrected gene. It also, as I said, you can, in Cas9, you can replace the nuclease with virtually anything you want, and they actually replace it with a reverse transcriptase. So you have reverse transcriptase and the RNA that is going to correct, going to be used for homologous recombination, but they're delivered simultaneously to the targeted gene. It's reported that much higher level of success and fewer off-target effects by doing this. Techniques are still evolving using CRISPR-Cas9. There's lots of different techniques being used. They're trying just about everything they can to exploit this miraculous tool. As you may or may not be aware, there is a huge patent fight over who gets to patent CRISPR technology. It's huge because you're looking at millions, if not billions of dollars of revenue a year. Yes, higher level of success by delivering a RNA that contains the corrected sequences that you want, as well as the guide RNA that will target the gene. And so you've delivered, after reverse transcriptase, you've delivered the double-stranded DNA that will hopefully, through homologous recombination, replace the existing gene that was cut by CRISPR-Cas9. Okay, that's it for HIV. We're not going to go into any of the other stuff. Now we're going to start talking about cancer. We won't get through all of it today. We will continue it on Thursday. We've talked a little bit about cancer, but we're going to go into some greater detail now with this presentation. Cell cycle regulation and the genetics of cancer. Homeostasis. If you think about it, our body is in constant surveillance to ensure that we have the right number of the appropriate cells. Not too many, not too little. It's critical. Maintenance of proper cell number in the immune system, in every system you can think of. If you have too few cells, you stimulate cell replication to repopulate. If you have too many cells, typically you initiate apoptosis. And we're going to talk a little bit about apoptosis. What happens when the system gets disrupted? Well, oftentimes it results in cancer, uncontrolled growth. So cells are constantly balancing between going through the mitotic cell cycle or committing suicide, apoptosis, if there's too many. Apoptosis can be programmed during development, such as the cells that are originally present, giving us webbed hands and feet. Those cells are developmentally programmed to undergo apoptosis. So we don't end up with webbed hands and feet. There are some mutations out there where people actually do have the webbing present. Might help them swim, I don't know. So you have programmed cell death, apoptosis, as part of normal development. Apoptosis can also be initiated in response to some abnormality. Too many of a cell type, they can't sustain the large numbers, they commit suicide, undergo apoptosis. Cells infected with viruses also can commit apoptosis to try to halt the spread of the virus. Most cells in our body are primed to commit apoptosis. They need an external signal in order to not undergo apoptosis. So the vast majority of our cells are just waiting there to commit suicide. As I say, they need some kind of external signaling in most cases to remain viable and not start to undergo apoptosis. Stem cells, we haven't really talked about very much. Stem cells are undifferentiated cells. Cells go through several different developmental stages. Determination is where they get their instructions as to what type of cell they're supposed to come. Differentiation is when they actually become that cell, when cells faded to become muscles, cells actually do become muscle cells. So that's when differentiation occurs. Stem cells are undifferentiated cells. The ultimate stem cell is the fertilized egg because everything is going to be derived from that. But as development proceeds, most stem cells have a limited potential future. That is, they can only produce certain kinds of cells. They can't produce all kinds. So they're restricted in the potential fate that they can undergo. Surveillance is the term we use to talk about what our body is constantly going through, monitoring levels of cells to ensure that we have the right number, not too many, not too few. Too few, mitosis; too many, apoptosis. Typically, what happens is the stem cell is stimulated to go through the mitotic cycle, and you have an unequal division. That is, the two daughter cells are not the same size. One, the larger one, is going to remain undifferentiated. It's going to remain a stem cell. So you're constantly repopulating your population of stem cells. You don't want to run out of your stem cells. You never want to deplete the stem cell population. The smaller cell is the one that goes on to differentiate, becoming a white blood cell or whatever its fate is determined to be. So the larger one remains a stem cell. The smaller one differentiates and takes on its fate. Surveillance typically involves signals from adjacent cells, intracellular signaling pathways communicate this, prevent cells from undergoing apoptosis. So you have a signal, some mechanisms for receiving that signal, typically a receptor, and then transduction or movement of that information from outside the cell to inside the cell. And that can involve things like cell movement, cell migration, can involve turning on transcription factors. Several different things that can be initiated by this cascade of events. Cell cycle. M is for mitosis. G1 is a preparatory phase, preparing the cell for DNA synthesis, as well as the histones and everything else that's required to make a fully functional daughter cell. The G0 phase of the cycle is for cells that are not going through the cell cycle. It's an arrested stage. They're not preparing for mitosis. They're simply existing. S is for synthesis. It's where DNA synthesis, histones, other proteins are produced. G2 is another preparatory stage, preparing the cell for undergoing mitosis, splitting of the nucleus and the cytoplasm. CDK, cyclin complexes, drive the cell cycle. They turn on transcription factors, amongst other things, express genes like DNA polymerase, production of the nucleotides. The deoxynucleotides are going to be required for DNA replication. Other proteins necessary for chromosomal replication, such as the histones. And it produces the next CDK cyclin in the complex. So there's a series of these proteins, CDK cyclin, that are turned on at consecutive steps in the process. There is a consensus sequence shown here. The main thing is it's a serine or threonine targeted for phosphorylation. It was discovered, we talked about a little bit, using temperature-sensitive mutants in yeast. What they did was to grow the cells, the yeast cells, in a permissive temperature, or typically a lower temperature, They would add a mutagen. They would then plate these cells out at a density that single cells would form discrete colonies on the Petri dish. They would then make a replica of that. They would literally take cheesecloth and quickly place it on the original Petri dish, go to another Petri dish and have a perfect imprint of the original one with all the colonies in place, so that you could then go back and identify the colonies that you're interested in. You would leave one at the permissive temperature, in this case 22 degrees centigrade, roughly room temperature, and the other one you would place at a higher temperature, 36 degrees centigrade. And then you would look for colonies that grew at 22 degrees, but not at the higher temperature, not at the non-permissive temperature. That would tell you that those cells had a defect in cell replication. You can get an idea of the sequence of these different genes by making matings between them. You can see that the first strain, CDC7, arrests in the bud stage, where the cell has started to divide but hasn't completed division. The other mutant, CDC28, doesn't even make it to the bud stage. And that tells you then that CDC28 actually occurs earlier in the cell cycle than does CDC7. So they could determine the sequence of expression of these different genes. These proteins, the CDKs, are remarkably conserved. You can take mutants in yeast, where you've knocked out one of these CDKs, and give it the human version and affect complete rescue. Very highly conserved proteins, which is not surprising. The cell cycle is a very old system, evolutionary. This just gives you some illustration of the sequence of some of the CDKs, where they're occurring. G1, end of G1, in S, and in G2. Don't expect you to memorize these. Again, the numbers as well as the letters really primarily signal the sequence with which they were actually initially isolated. They don't really tell you anything about which comes first and which doesn't. This is basically the system. It involves a kinase as well as a phosphatase. While the CDK is phosphorylated, it remains in an inactive state. It's not going to allow the cell to progress through the cell cycle. Introduction of a phosphatase that removes the phosphate from CDK, converts it from inactive to active, and now it can stimulate the cell to progress through the cell cycle. In this particular instance, it was moving into mitosis. So it's the interplay between the kinase and the phosphatase that regulates all of these genes. As I think I told you, this work was done in yeast. At the same time, we had a colleague who was studying human eyes, development of human eyes, who went around telling all the graduate students he had no idea why anybody was going to study anything in something that wasn't a human tissue culture system or somehow directly connected to humans, including mice and Drosophila. He was very narrow-minded, did not have very much breadth. At the same time, these investigators in yeast got the Nobel Prize for understanding the cell cycle. Okay, now we're going to talk about a very well described protein, retinoblastoma, that is involved in cancer formation. This is my attempts at illustration. The cyclin A, CDK2, as long as the phosphate is present, is inactive. Once the phosphate gets removed, what happens is you formed an active complex. RB is a retinoblastoma protein. E2F is a transcription factor. The function of retinoblastoma is to tie up the transcription factor, preventing it from migrating to the nucleus. When there's an active cyclin CDK2 complex, it's going to phosphorylate the retinoblastoma protein, and in phosphorylating it is going to cause the release of the E2F transcription factor. It can now migrate to the nucleus and initiate gene expression and movement progression through the cell cycle. Another illustration. You can tell I'm not terribly good with trying to draw figures in PowerPoint. As long as the retinoblastoma is phosphorylated, it's inactive. It cannot bind to the E2F. Loss of the phosphate, through the use of a phosphatase allows E2F to now be released and go to stimulate S phase of the cell cycle. Retinoblastoma is part of a group of genes called tumor suppressor genes. These are genes that, as long as you have one good copy present, the cell will not go to become cancerous. So one copy is sufficient to suppress the formation of tumors. So it's a recessive in terms of inducing cancer. It's a loss of function type of mutation. Both copies have to be mutated in order for the cancer to develop. Come in basically two different types. They're negative regulators of cell cycle, like the retinoblastoma. As long as you have one good copy of the retinoblastoma, it's going to tie up the E2F factor, preventing it from indiscriminately migrating to the nucleus and initiating transcription. Can also be an indirect. It's positive regulators of apoptosis. That is, genes that are required to induce apoptosis. And you lose the ability to induce apoptosis, surveillance goes out the window, cells that shouldn't be dividing suddenly are dividing, resulting in cancer. So two different ways. One is a negative regulator of the cell cycle, the other is a positive regulator of apoptosis. You can also have indirect action, genes involved in DNA repair, for example, or genes involved in detecting defective cells. Those of you that are unfortunate enough to have suffered from a sunburn, that is the body's defense mechanism telling the cells that are too damaged by UV radiation to commit apoptosis, which is why you get your skin peels in a bad sunburn. Okay. Retinoblastoma is a cancer that's caused by the loss of both alleles of retinoblastoma. It's typically a childhood cancer. It's a cancer of the eye. It takes two different forms, one of which is sporadic, meaning it just arose. It's not inherited. And the other is familial, indicating an inherited predisposition to develop retinoblastoma. In the case of sporadic, There's no family history of retinoblastoma, and typically it's relatively minor. Individuals with this cancer typically will lose vision in the eye in which it arose, but the other eye is going to be perfectly okay, and the incidence of other cancers is going to be that of the normal population. It's a somatic mutation, soma, non-germline. In the case of familial, there's a family history of ancestors having retinoblastoma. This is by far the more serious situation. Typically, tumors develop in each eye, and there's a 6-fold increase in other forms of cancer as well. So retinoblastoma, the protein, is named after the cancer of the eye, but it also plays a role in ensuring E2F does not migrate in other cell types as well. So the familial one is pretty devastating. Again, that's where a pedigree analysis comes in very useful. Considered, as I said, tumor suppressors. The two-hit hypothesis. In order for cancer to occur, you have to knock out both versions. In the case of familial retinoblastoma, the individual inherits one defective copy. So there's several different ways that the good copy, the wild type copy, can be lost, resulting in homozygosity for retinoblastoma mutation. And they inherit one mutant allele. It predisposes them to development of retinoblastoma. It doesn't guarantee it, but it happens in a fairly high incidence. As I say, it requires the loss of the second locus, the wild type copy. Lots of different ways it can occur. Mutation of wild type copy or loss of heterozygosity, LOH. We don't talk about mitotic crossing over. We talk about meiotic crossing over during meiosis. But the truth is, you also have mitotic crossing over that can occur. You can have non-disjunction, loss of the chromosome that carries the good allele of retinoblastoma. You can have gene conversion, deletion, point mutation, several different ways that it can be lost. And here's an illustration. This is a young female with the eye on the right. She has a retinoblastoma present. This shows you the genetics behind it in the pedigree. One parent was a carrier, and the child received one mutant copy and one wild type copy. Over time, the wild type copy is lost, either through non-disjunction, and replication of the mutant-bearing chromosome. You can have mitotic recombination occurring, eliminating the wild type copy, homozygosing for the mutant one. You can have gene conversion, which is an attempt to repair DNA using the complementary strand from the other chromosome to repair. And in doing so, again, homozygous for retinoblastoma mutant. You can have a deletion of the wild type copy, or you can simply have a point mutation, converting wild type retinoblastoma to mutant retinoblastoma. All those different ways can result in inability to suppress E2F, resulting in different cancers, especially the retinoblastoma. Okay, on the right, we have cancer cells. Cancer cells often can autocrine stimulate, that is, they produce a molecule, a ligand, that will interact with their receptor, stimulating mitosis. Wild type cells, non-cancerous cells, exhibit something called contact inhibition. If you plate them out on a petri dish, they will not grow over top of each other. They communicate and do not grow on top of each other, as I said. Cancerous cells have no inhibitions. They grow over top of each other all the time. It's probably the best way to tell if a cell is cancerous. Radiation, normal cells are highly susceptible to death caused by radiation. Cancer cells, very resistant to death induced by radiation. Normal cells have gap junctions, which is how they communicate with each other. It's how they know not to grow over top of each other. Cancer cells do not have gap junctions. They have no ability to communicate with each other, which is why cancer cells grow over top of each other. And normal cells, wild type cells, non-cancerous cells do not. Another way, another kind of origins of cancer are so-called oncogenes. The tumor suppressors, you have to have homozygous mutant, so they act as a recessive. Oncogenes, on the other hand, are actually dominant. Tumor suppressors are loss of function, LOF, while oncogenes are typically a gain of function, or GOF. The gene has required new characteristics. Usually oncogenes are an overactive protein which promote cell growth. How to identify oncogenes? A couple of different ways. Because they're a gain of function, as I said, there are two approaches. One of which is to look at tumor viruses, viruses that you know cause cancer. Feline leukemia virus, for example, is one. I actually worked in an RNA tumor virus lab for about 2 years, and we would grow tumor cells in 20-liter carboys, huge bottles, if you will, that we would grow as tissue culture, and then harvest the viruses from the tissue culture as they're released to the media. Initially, hadn't figured out all the conditions required for being able to grow some of the viruses in tissue culture. And there was one in particular that we were interested in investigating. It caused mammary tumors, breast cancer. It was a virus that infected mice called the mouse mammary tumor virus, MMTV. At the time when I was working there, they didn't know how to culture it in tissue culture cell. The only way they could get this virus was from the milk of mice with breast cancer. We had 2 people whose full-time jobs were to simply milk the mice. And they had little eyedroppers that they'd put on the teat of the mice and sat there all day getting little droplets of milk out of the mice. I often wondered how they described their previous job if they went somewhere else. Can't be too much demand for a professional mouse milker. Anyway, it was funny. I watched them do it one day. Very tedious. When an RNA virus inserts, oftentimes it will insert near a proto-oncogene, a gene that is capable of giving rise to this dominant phenotype, gain of function, and it will oftentimes capture the adjacent gene and carry it with it when the virus replicates. And so you look for genes that shouldn't be present, that need not be present in the virus. Typically, the viruses have their own promoters that run at a very high level, so they're upregulating the expression of this proto-oncogene. The oncogene, as I said, gets captured lots of times by the virus. And so you look for genes that are not normally part of an RNA tumor virus. There are 3 genes that are commonly found: GAG, POL, and IMP. These are the normal genes found. If you find another gene and you know it's an RNA tumor virus, odds are really good that that gene is an oncogene. Any other way? I told you there was 2 ways. Sometimes I don't lecture on this and I actually make it a question on the exam. Not this time, at least not as I'm presenting it now. How could you identify a gain-of-function mutation? A hint? Think back to early evidence for DNA, in particular pneumococcus, transforming factor. You can take cancerous cells that you know, because of their behavior, are acting like a dominant gain of function, and you can extract the DNA and transform normal cells to now become cancerous because it is a dominant gain of function. You take DNA from human tumor cells and mix with normal, that is non-cancerous, mouse cells. The mouse cells that take up the human oncogene will now be cancerous. They will have lost contact inhibition. They will grow over top of each other. So now you know population of mouse cells have taken up the oncogene. But how can you tell the oncogene from the mouse's DNA? Examine mouse cells that lose cell inhibition, cell contact inhibition. Well, talked about ALU, how it's a degenerate transposable element. Relatively short, 200 to 300 base pairs in sequence. Millions of copies are found in our genome. The odds are really good that in addition to the oncogene, the virus has picked up an ALU sequence. And the nice thing about ALU, it's only found in primates. It is not found in mice. So you can screen a library of mouse DNA using ALU as a probe, looking for segments that have taken up human DNA, and clone that, sequence it, and determine what the oncogene is. So 2 different mechanisms for cancer. One is tumor suppressor, where the mutation has to be homozygous, so it acts in a recessive, loss of function. And the other is a gain of function, the oncogene, where a gene has taken on new properties and can cause uncontrolled cell growth, cancer. One of the more famous examples is a hybrid between the BCR gene and the c-Abl. C indicates it's the cellular version of a gene found in an RNA tumor virus, Abl. The oncogenes are normal genes required for normal development that somehow something has gone wrong with their regulation. This one, it's a translocation, results in chronic myelogenous leukemia. It's a fairly common form of leukemia. It's a translocation between chromosome 9 and 22, so-called Philadelphia chromosome, because it was discovered in the city of Philadelphia. Fuses c-Abl with BCR. c-Abl encodes a tyrosine kinase. Not surprising that it would be a kinase. In its normal situation, not in the translocated version, but in the normal location, its expression is regulated by growth factors. In the translocated gene, it's not regulated. It has lost its ability to be regulated, and as such causes cancer, unrestricted cell cycle. It's always active. Drug companies, this is a good example of why we should invest in cancer research. Drug companies had isolated, or universities, someone did, isolated the abnormal protein, the product of the translocation, and they were able to get X-ray crystallography. Remember we talked about X-ray crystallography in terms of understanding DNA structure? Well, it can also tell you about protein structure. And so they had crystal structures. They knew what the protein looked like. c-Abl, which is a kinase, binds ATP to get the phosphate. Phosphate has to come from somewhere, it comes from ATP. So you have a binding site for ATP. The normal cellular protein as well as the fused protein both have to have a binding site for the ATP, otherwise they would be non-functional. Gleevec, I think is how it's pronounced, was designed to fit into c-Abl's ATP. So it binds, it competes for binding on the protein. Prevents ATP binding. In the absence of ATP binding, you're not going to get a phosphate group added. In clinical trials, 98% of individuals with the Philadelphia chromosome, this translocated tyrosine kinase, saw complete disappearance of the leukemia. And 5 years later, which is often the benchmark for successful treatment of cancer, only 5% died, whereas the normal lethality would have been 50% or more. So a designed drug that works to reverse the effects of what would otherwise be leukemia. Not all oncogenes have been targeted. The real challenge in doing X-ray crystallography is to actually get the crystalline structures. It can be a real challenge. But this is an example of a success story. Here we have the translocated protein, the fused protein, with a kinase domain. The gene binds ATP and can now phosphorylate targeted proteins, causing them to go through the cell cycle. And this chemical, another one by another company, 2 different names, no idea where they get the names from, actually binds to the site where ATP normally binds, blocking the substrate from binding. Cells can't proliferate, and ultimately they're going to die. There are 3 known non-viral infectious cancers in the world. There may be more, but there's only 3 that are known. These are cancerous cells that are transmitted and establish new cancers in their new host. So the cancerous cells that become transferred actually establish themselves and cause cancer. One is in dogs, and it's sexually transmitted. Fortunately, it's not very common. Another is seen in West Coast clams. It's a bit odd. And probably the most studied is in the Tasmanian devils. if you watch cartoons when you were growing up, you should recognize the creature on the left, holding down Bugs Bunny in the bottom picture. This is what the Tasmanian devils actually look like. They're tiny. That's a 20p coin, British coin. They fight constantly by biting each other. And what happens when they're biting each other is that's when the cells get transferred. The cancerous cells get transferred to wounds inflicted upon the recipient. They're not very big, but they apparently do fight like devils. You can see they got lots of nice sharp teeth. And they are cute though. These devils, as I say, the cancers are transmitted through bites, when they bite each other, when they're fighting for mates and whatnot. They bite each other on the face. That's where the cancers get established. Results in facial cancers. In 20 years, the population, the wild-type population, has declined by 80% because of this infectious cancer cell. Fortunately for the Tasmanian devils, they've been rapidly evolving resistance to these cancerous cells. Drugs that were developed for humans to treat cancer in humans have been successfully used to treat the Tasmanian devils. And it looks like they've actually sort of turned the corner. The drugs are targeting RTKs, receptor tyrosine kinases. You see that this is a very common theme throughout development, is the role kinases and phosphatases play in the process. And then this is another example of a cancer that's caused by runaway tyrosine kinase activity. Proper coordination of the cell cycle is critical, going back to some work we talked about a little earlier. Proteins must be produced and become functional at the proper time. By the same token, the cell has to complete one phase of the cell cycle before it can move on to the next one. And so not only is expression critical at the proper time, but turning it off is also critical. You don't want a protein hanging around that's going to cause the cell to repeat the same cell cycle it just went through. Checkpoints ensure the cell is ready for progression through the cycle. So it goes through a series of checkpoints to determine, okay, looks like everything's good. We can go through the cell cycle. DNA damage. Again, skin cancer, which I've had, by the way, 12 different times. And I've had all three forms of skin cancer. Melanoma, which is the most deadly. I've had it once. The other two are not. Squamous cell will metastasize, but if caught early, as with melanoma, it's very treatable. And then the most common form, which is at epidemic proportions in Australia, is, I can't think of the name now. It's epithelial cells. It also, basal cell, that's the term. They're all named after the cells that become cancerous. Basal cell is malignant. The cancer will grow, but it grows very slowly. If it's in frustrated with your student health. For about 3 years when I was in graduate school, I kept going to UVA student health and complaining about this sort of sore on my right arm. They kept giving me ointments for my skin, like it was a rash. I did my postdoctoral fellow at Fox Chase Cancer Center. Everyone had to have a physical before you started working there. As soon as the physician's assistant saw the spot on my arm, she said, "Oh, you have basal cell carcinoma." Great. I'd had it for like 3 years or so, and it had grown, but very slowly. It started off the size of a pencil eraser, and in 3 years, it had grown to about the size of a penny. It can metastasize, but it's extremely rare. It almost never metastasizes. It does need to be removed. It can grow on the surface of the skin, but it can also penetrate inside, grow downwards. And in doing so, can disrupt muscle, bone, nerves. And when it starts growing downward is when it has the potential to metastasize, but it's extremely rare. And it is never included in calculations of cancer survival because it's such a rare event that people let it go for way too, we're talking decades before it has the potential to metastasize. So, and that's been the majority. I've had all three. The vast 8 out of the 12 have been basal cell. 3 were squamous and 1 melanoma. Fortunately, the melanoma was in stage 1, so very treatable. DNA damage. There are proteins to ensure that cells that have undergone DNA damage, such as through sunburns, do not progress through the cell cycle. Instead, if they cannot repair the damage, they undergo apoptosis, which is why your skin peels if you get a severe sunburn. The cells are doing what they're supposed to be doing. They're committing suicide rather than going on to cause cancer. I personally think tanning booths should be outlawed. I mean, it's bad enough we allow people to smoke cigarettes, but allowing people to knowingly subject themselves to a cancer-causing source of radiation should be outlawed, in my opinion. But my opinion is somewhat clouded by my own experience. Typically, once every 6 months, I go to see a dermatologist. He uses liquid nitrogen to freeze off precancerous spots. And usually, every time I go, I've got anywhere from 20 to 30 spots that are precancerous lesions, mostly on my head. And I always try to schedule my visit late in the day because it gives me a mild migraine, all the liquid nitrogen being applied to my scalp and face and whatnot. I hate it, but it's the penalty I pay for having spent most of my childhood outside playing in the sun. When I was young, sun lotion was intended to enhance the damage caused by the sun. Sunscreen was unheard of. And again, when I was growing up, they had 3 stations, NBC, ABC, CBS. All of them during the day had soap operas on. And at least none of the kids I knew my age wanted to stay home and watch soap operas. So if the weather was nice, you spent all day outside. The only time you stayed indoors was when it was raining and you really couldn't go outside. And that's when we had things like stamp collections and coin collections and reading of comic books, which is why comic books are so big with my generation, because that's what we did when the weather was bad, but resulted in a lot of exposure to the sun. Okay, P53 is one of the checkpoints. It's mutations of which are commonly found in cancers, tumors. It's a checkpoint that ensures cells that have undergone DNA damage does not progress through the cell cycle. It gives the cell a limited amount of time to try to repair the damage. And if it can't do so, it instructs it to commit suicide, to undergo apoptosis. P53 minus cells have lost this checkpoint, and cell replication can occur unchecked in terms of damage to the DNA. Now, you have to have something that stimulates it to go through the cell cycle. The loss of P53 alone is not sufficient to initiate formation of a cancer, but once it has initiated, it will continue because you don't have one of the critical checkpoints. Okay, I think we'll call it quits today. We'll pick up and finish this chapter on the genetics of cancer. I'm going to be talking about an inhibitory control of progression through the cell cycle when we meet again. Again, I hope to get all of the recordings posted to YouTube, and I will be sending out an email once this has been achieved. Stay healthy, stay safe, and we will reconvene Thursday. Bye, folks.